I'll introduce myself. My name is Sam Lawrence. I'm the CEO of CytoViva. And I want to cover today a overview of the CytoViva system that is now there on the campus. I'm going to focus mainly on kind of how it works up front, and then go into some applications around drug delivery and discovery and exosomes. And then open it up for questions. Since there's only a few of us here, we can be pretty informal as we go through this. And I will start by sharing presentation, CytoViva has based on two key components. One is a enhanced dark field optical microscopy illuminator, which allows the rapid observation of nanoscale materials with no sample prep required. And the second is a hyperspectral imaging system, which allows you to capture spectral characterization of nanoscale entities based on their surface chemistry and map them in complex matrices. This is what the system looks like. Starts with a light source, 150 watt halogen light source, and then that light is then projected into an enhanced dark field optical illuminator via liquid light guide, which then creates an image. The motorized stage moves and allows the spectrophotometer to scan that image, creating the optical and or hyperspectral image that you see there on that left screen, which then you can then go and query at an individual pixel level to get a full, in this case, visible near infrared spectra from 400 to 1,000 nanometers of curve, representing the spectral response of that individual pixel. So, some applications and the kind of places that this has been used before, on the left column there, you see sort of government agencies, FDA and nanotoxicology, NIH for nanodrug clinical trial testing. In the middle, you see, research institutions like IIT Madras, nanomaterials development, Duke doing environmental talks. And then on the right, you see private companies, Merck doing nanomedicine and Novozymes doing biofuels. All that's just to show that there's a wide range of things that we can do with this. And so I'm going to focus today on nanodrug delivery and exosomes. But if you have, or any of the people that you in your ecosystem that you work with on a daily basis, have questions about other applications, including those that are reflected light, can you do hyperspectral imaging on reflected light applications? Please reach out to us, and we can talk to you about how to do that. It all starts with an enhanced dark field illuminator, which takes, as I mentioned, light from a light source through a liquid light guide into a series of collimating lenses, where the light is then projected onto a mirror and up until the annulus of the dark field condenser, which then concentrates the photon energy onto the sample on the slide, thus projecting maximum photon density off the sample into the objective and allowing you to create the nanoscale imaging. This results in a 10x improvement in signal to noise over standard dark field microscopy, which is really important when you're trying to do hyperspectral imaging, because the strength of the signal is going to do a whole lot to determine the quality of the data. So as you can see here, this is a test done in one of our clients in Korea. They used standard dark field on the upper images there on a 160 nanometer magnetic nanoparticle, and then CytoViva on the one below. And you can see that the much clearer signal and about a 10x improvement in signal to noise. So the hyperspectral piece is pretty simple. It consists of, as I mentioned, moving the stage underneath a visible near infrared or shortwave infrared diffraction grating based spectrograph. You guys have the visible near infrared and integrated detector. That is then capture the data line by line, kind of a push broom scan, to create a data cube, which allows the the user to go in and query that data cube at the pixel level. So you're measuring unique reflected spectra again, either in the visible and infrared or shortwave infrared, 900 to 1700. I will mention that that does require a different detector and spectrograph than you have currently. You get the full spectral profile for each pixel. Pixel size on the actual sample can be as small as 128 nanometers of space on the sample at 100x. At 60x, which is sort of the mag that we use most, it's around 200. The spectral data is recorded in very high resolution, down to 2 nanometers, and then represented as both a spectral curve and as an RGB image separately, as I showed you in that first picture. So you have a visual reference of where to go and query. These two things together create the ability to get very high, very precise data on individual nanoscale materials. So the image on the left is standard 100 nanometer gold nanoparticle imaged with standard dark field microscopy. The image on the right is that same nanoparticle imaged with CytoViva. So below that, we scanned it and created a data cube, and then we go in and queried the pixel that's the brightest in each image. And as you can see below the first image, you see a very choppy signal with low amplitude, and as compared to the one on the right, which is much cleaner with a much larger amplitude. But even more importantly is where is the peak located? So with a 100 nanometer gold particle like this, we know that the peak location should be around 650 nanometers, which it is here in this image. Over here, it's around 600, and that's because the data is so bad that you're not getting a good clean signal. So when you're doing hyperspectral microscopy, you need that high signal to noise imaging capability to get good quality data. So that's the basics of how the system and the images are created. Here's some examples of just basic capabilities and what that results in your work. These are gold nanoparticles up top aggregating the water, and then they're buffered below in an ionic solution. So as you can see in the upper image, they aggregate and form all kinds of different shapes and sizes. And that's represented by a whole variety of curves just randomly selected over this field of view. However, when you buffer them and they disaggregate, you see the individual particles who are very consistent. And as you can see, when you go in and image and collect data off of each of these particles, how the data stacks up right on top of each other. So that kind of speaks to the consistency of the data available through the system. Secondly, as I mentioned, changes in surface chemistry. So here you have 3 different samples. The first 2 are 200 nanometer gold shells, bare. And the spectral data from an average particle here is represented by this green and red curve. In the middle, you have those same 200 nanometer shells coated with PEG. So just by changing the surface chemistry, even though you have the same base particle, you get a very different response and so that, as you can project that forward, you can see how that would be useful in doing drug discovery work as you functionalize particles. So I'll show you a real quick environmental tox example because it shows you kind of the workflow of the system. So here you have an algae cell that's been exposed to silver nanoparticles, and you image the cell and you create a spectral library of the silver particles in that cell. And then you can go tell the software to map those particles where in the cell, okay, based on that spectral library. Now, we have built several different techniques to help facilitate this, including the ability to filter spectral library, meaning that you would take a control cell, and filter this spectral library against that control to avoid getting false positives. There are other techniques that we can show you in the training. Furthermore, though, that library is different for a nanoparticle that's inside the cell, such as this one, versus one that's on the cell membrane interacting, such as this one here. So that again speaks to the sensitivity of the technique and the kind of results that you can expect to get in your work. So let's talk a little bit about measuring drug loads and functional groups on nanoparticles. Here, I'm going to give you some examples, just several in the drug discovery realm. The first is, loaded liposomes loaded with gadolinium. Here is the loaded liposomes here imaged at 100x, 60x. Here below, now optically they look different, but spectrally you can see they're very different because here you have the curve of the liposome represented here, and then you have here with the loaded one, you have the influence of the gadolinium here out at 770. Similarly, loaded and unloaded polymeric nanoparticles, these are the unloaded ones in the white versus the thrombin-loaded polymeric nanoparticles in red. So again, you're changing the surface chemistry, you change the way the light interacts, and therefore the optical spectral response that you get from each of those particles. You can run statistics on these particles and get regression statistics. You can also, when you map them, you can get data to say how many pixels in the whole area of the image mapped as a percent. Here we have polymeric particles, again with the drug load, and then one with a bare, and then one with a API. And you can see not only are they different visually, but you also can map the API on the nanoparticle itself. So you have a different spectral signature, unloaded versus loaded, and then you can locate where the API is on the particle. This is a very typical example here. You have macrophage cells exposed to LDLs loaded with gold nanoparticles. So, you have your cells, and you want to identify the LDLs and map them. So this is the cell structure, this green, kind of a mean curve for an unloaded area of the cell versus the loaded LDL loaded gold particles. Okay. You can then tell the software to go map those, and it finds where they are, and then we'll give you statistics on how many particles mapped as a percentage of the total area. Again, you want to always run these kinds of experiments against a negative control sample to map filter your spectral library so you don't get false positives. And we made that pretty easy in customizing the software. This is charts just to show you that this was something the FDA presented, probably like 6 or 7 years ago now, from to demonstrate, Catherine Tyner's lab, to demonstrate that CytoViva was a useful tool in working through, especially the CMC section of drug approval, as it was one of the few things that could actually show researchers where the nano drug delivery vectors went inside individual cells in a non-destructive manner. Again, you don't have to label the cells you don't have to label the particles or the cells to do this work. So let's look at some exosomes. So these are 3 different types of exosomes. Okay. And I can't tell you much about where they came from, except just to tell you that they're 3 different types. So even though they're all exosomes, they all have different spectral signatures. So you can imagine that you can map them differently in an individual in the sample if they were together. Okay. So I'm going to show you real quick. I'm going to open up a data cube and show you what that looks like. So So this is what sample S2 looked like. And these are those exosomes imaged optically and scanned hyperspectrally. So that's this is the results you would get after you scan them. Then you can go in and look in a zoom window. So you see this zoom window here on the right. And this is the area where we're looking. And you can zero in on an individual particle and collect here the spectral profile of that particle. Similarly, you can click on a different one and get the different spectral profile. And you can look at these different particles and you can see they may have different amplitudes, but their basic shape is the same. So you can then go validate that, let's say that these are the same particles and you want to go map them. You can build a library to go map them. And as you can see, they pick up the components and they turn red. Okay. So that's just a quick example about how the software works, the kind of data you see when you open it up, and how you can build a library and do mapping. So a really quick and dirty example. So now, going back, other oops. So these are exosomes again. Sample 1, sample 2, two different types of exosomes with two different types of spectral curves representative. And that again showing you that even though they're both exosomes and they may look very similar, their protein structures will give you different reactions. Okay, so here's an example of bacteria and exosomes interacting with each other. So you see the exosomes only by themselves, these little blue dots, these are the bacteria, and there's some biofilm here. And then this is the bacteria and the exosomes interacting with each other. So you can then go create a spectral library of the bacteria by themselves. That's the control. And then you can get libraries of the exosome by itself, and the two of them interacting with each other, with the exosome in the bacteria. So you can see the exosome attached here, and then this is the control with no attachment. And you can go map those things, as I mentioned before. So then here's some live epithelial cells exposed to exosomes. Just a basic epithelial cell. This is the cell with the exosomes. So the bare cell had this response. The exposed had the green response, and they are different. And then you can go map them, the interaction in the cell. And then finally, some human vein epithelial cells, human cubic cells. So here, these are the EVs as they shed from the UVEX. Okay, and this is spectral curves resulting from those EVs. And then this is the image of the UVEX themselves with the endosomes prominent in the cell culture, and then going and mapping them in those cells. So those are some really quick examples of both drug discovery and EVs. This is one of the things I also want to share with you. You don't have this equipment on your system, but the 3D imaging that's available to help with drug discovery, locating nanoparticles in cells. The images I showed you before, as you can see, that when you look at the cells, you can see that you could you're looking at a 2D image, right? And you can focus up and down through the focal plane to see where the particles may be. But if you could create a 3D representation of those cells, it would be super helpful. So we built the ability to do that. So what this does is it takes that 2D image and we collect a stack of images. And then we locate the particles in a 3D space and deconvolve the cell structure, both the unstained, which is represented in gray, as well as any stained structure, which in this case is represented in blue. We deconvolve those and then we push those all three together in a 3D viewer in image J. And that allows you to zoom in, zoom out, turn it, and see, in this case, these are called nanoparticles, and see where they're located in that 3D space. This is actually some work done by Devika Chithrani before she went to UVic. She's imaging some, again, gold nanoparticles in a DAPI stained nucleus with GFP color stained or tagged drug load in the middle. So as you can see, the GFP kind of poking through there in the middle, it's actually penetrated into the nucleus. And she was able to demonstrate that with this technique. And then you can take different channels, if you will, out, and you can see the GFP there as you took the DAPI stain channel away. Finally, I wanted to share with you that we do have the ability to combine our system with the Raman multimodal system from Hariba, the Explora Raman microscope. So we can combine our hyperspectral equipment with their Raman microscope. So you can do studies where you are cross-validating the CytoViva results with the quantitative results from Raman. This is an example where a user was locating ceria and aluminum oxide in cells. So this shows you here, what it would look like under bright field, and here's what it looks like with CytoViva. So you can actually see the ceria particles. You scan them, create a map, a library, and then you can map those ceria particles in the image. Then you go back and cross-validate the results of that scan with Raman to get the specific Raman signature from these ceria particles. And you can do things like, you can do that with, gold particles with the SIRS activation as well in tissue and other things, polymeric particles, etc. So I wanted to share that with you as well. So that's really all I had to share in terms of kind of how the system works, and I kind of want to open it up now and give you guys some opportunity to ask questions. So Joel, Shadar, Aaliyah, any questions?
Yeah, I was kind of wondering, like, you showed those nice images with like the cells, things like that. I was curious if you've done any of this with kind of like 3D cell cultures or like matrigel, or kind of, maybe well conditions or play conditions that might have higher like background signal. And I was wondering if you've had any kind of like signal to noise issues with that.
Yeah, so to get the response that you're seeing here, you really do need a monolayer of cells on a slide. If wells have a thick bottom and they're really difficult to image through. Now you can always do this in reflected light, although I don't believe you have a reflected neck on the system there that you currently have at the pharmacy. You can add one of those relatively easily. It's not super expensive to do so. But if you want to do imaging like that, I would typically say you need to do it from a reflected light modality. It does not work that well in the transmitted enhanced dark field modality. So you lose some of the nanoscale clarity, if you will.
Got it. And for those cell images, those were all like fixed cells, I assume?
Yeah, yes. So either cultured, on a cover slip or fixed to a slide.
Okay, perfect. Thanks.
Mm-hmm.